Welcome back, everyone. I now have the distinct honor of recognizing a milestone in our incredible NYSCF Robertson Investigator Program. I want to first thank the Robertson Foundation for their pioneering support and visionary partnership in creating this extraordinary program with us. It's so meaningful to me, to NYSCF, and to patients around the world whose lives are already being improved directly as a result of the NYSCF Robertson Investigator Program. I also want to extend thanks to all of the NYSCF staff, and in particular, Kristen Smith, who has worked tirelessly on the program since its inception 10 years ago. As I reflect back on the last 10 years, I am in awe of the tremendous progress that we've made and the huge impact that the NYSCF Robertson investigators have already made on neuroscience and stem cell research. I think what's really been critical is the unrestricted funding, and it really has allowed these researchers to make paradigm-shifting discoveries and have across the board, the clinical strides that have been made date back to our very first class, Paul Tazar's work with MS, Marius Vernig's work with EB, Shuibing's work now most recently in COVID-19, and Derek Rossi with hematopoietic stem cell transplants. So quantitatively speaking, these scientists really are second to none. 20 of them have received the NIH Director's New Innovator Awards. 5 received Presidential Early Career Awards for Scientists and Engineers. 5 have received the ISSCR Young Investigators Award. And they were 5 of the last 6 Society for Neuroscience Young Investigators, as well as a Breakthrough Prize and many other prizes. The list really just goes on and on. But while their individual contributions and accolades are numerous, it really is the community, I think, that has made this group special. And excuse me, I think their truly collaborative spirit and the willingness to work together to create treatments and cures for patients is a very, very generous group. And fortunately for us during this pandemic, NYSCF Robertson investigators are always looking for the toughest challenges, the most important scientific challenges. And right now, COVID-19 certainly is at the top of everyone's list. And in just a few minutes, we're going to hear more from this incredibly innovative community about how the technology and stem cell research is being directed to take on any biomedical challenge, but in particular, the work that is being done in COVID-19. But before we get to the panel, now is a very exciting moment to bring up the new class of NYSCF Robertson investigators. As is our tradition, we have a distinguished alum announce the new class. And today I have the distinct honor of introducing one of our esteemed NYSCF Robertson investigator alums, Dr. Deepta Bhattacharya. Deepta is an associate professor of immunology at the University of Arizona. He truly embodies the innovator spirit that we're so proud of at the program overall. He is a profoundly creative scientist. He's an outstanding collaborator, including with NYSCF on our immunoengineering work. And luckily for all of us, he's currently doing important research on COVID-19, which we will hear about shortly. So Deepta, please join us.
Okay, thank you so much, Susan. I really appreciate that introduction. So as Susan said, my name is Deepta Bhattacharya, and I'm an alumnus of the NYSCF Robertson Investigator Program. I first found out I was receiving the NYSCF Robertson Investigator Award back in 2012 when I received a call in my office from a New York area code. And normally I have to say I don't answer my phone if I don't recognize the number, but this one time Maybe it was prescience, I don't know, but I'm so glad that I did. And it was Susan to tell me that I had received this really incredibly prestigious award. So, when I got the call, I knew that this award would change the course of my career. But in that moment, I really didn't understand exactly how transformational it would be. For me, the NYSCF Robertson Investigator Award has been an absolute game changer. I anticipated how huge of an impact the new funding would have on my lab, and especially the flexible funding that this award provides. It allowed me to pursue some big, bold, risky, and I think, one of the most important things is really long-term ideas that the current systems aren't really designed to support. And I think that's one of the key things about this award is that it supports and believes in the investigator. So that support comes not only from the funding, and as Susan alluded to, what I didn't fully appreciate at the time is just how much of a true community it is within NYSCF. The connections that I formed with other Robertson investigators, as well as researchers and staff at the NYSCF organization and labs continues to date and has really broadened the things I can do and has shaped my entire career. So that's why I'm here. That's why I feel so honored and compelled to be here and welcome these new investigators here tonight. Because these new investigators are about to join a program and a community that I imagine will change their careers too. So to the new investigators, congratulations. And let me offer just a little bit of a personal anecdote here. If you're all like me, I think you might have just a touch of imposter syndrome when you look at just these amazing investigators around you, both within your class and those that preceded you. And you may maybe wonder, like I did, I don't know if I can really hang with this group. But believe me, you absolutely belong. You are all standing here tonight because you're at the absolute top of your field. You're leaders and pioneers, and each of you has the potential to make huge contributions to your fields and significant positive impacts on human health. So I encourage you to take full advantage of your award and certainly of this community. So without further ado, it's my honor to welcome the 2020 class to the NYSCF Robertson Investigator Program. So I'll start with the 2020 NYSCF Robertson Stem Cell Investigators. And the first person that I'll be announcing here is Dr. Raffaella D'Amico. She's at the San Raffaele Telethon Institute for Gene Therapy in Italy. Let's over a little bit. Can we see her? Okay, there we go. Raffaella is working to optimize gene therapies that use hematopoietic stem and progenitor cells. Her work focuses on the biological impact of gene editing procedures on hematopoietic stem and progenitor cells for gene therapy applications in regenerative medicine, aiming for sustainable, safe, and clinically broad hematopoietic and stem cell-based therapies. Congratulations, Raffaella. The next person is Dr. Samantha Morris, who is at my former home and favorite place in the world, which is Washington University in St. Louis. Sam is interested in direct conversion and is leveraging genomic technologies to map the molecular changes of direct conversion and ultimately develop therapies to regenerate the small intestine and gastrointestinal disease. Congratulations, Sam. The next person is Jose Ordovas Montañez at Boston Children's Hospital. Jose is using stem cells to understand how and where disease is stored in a tissue, aiming to identify the root causes of inflammatory diseases in human tissues. He and his lab are working to show that the immune cells in the body are the exclusive source of memory formation in inflammatory diseases and are vital storers of disease states. Congratulations, Jose. I'd now like to announce the 2020 NYSCF Robertson Neuroscience Investigators. The first person I'll introduce is Jami Guo at the University of Calgary in Canada. Jami's research is focused on primary cilia, and she aims to investigate how primary cilia contribute to the brain's functional wiring and related disorders. Congratulations, Jami. Next up is Dr. David Schneider at New York University. David is working to understand how the brain stores and recalls memories to make and use predictions about the future. His research focuses on studying the interaction of auditory and motor systems during sound-generating behaviors to anticipate the sounds our movements make. Congratulations, David. And then our final Robertson Award recipient is John Tuthill at the University of Washington. John wants to understand the fundamental computations that underlie the neural coding of sensory information and to figure out how sensory signals are used to guide movement and behavior. Specifically, he aims to understand how and where motor circuits integrate proprioceptive information and why people without proprioception can execute some movements, but not others. Congratulations, John. And I don't see him. So he's presumably somewhere in the background here.
Great.
Okay, so thank you so much. And just let me again reiterate what a great honor it is to be part of this community. Congratulations again.
Thank you so much, Deepta, and congratulations to all of our newest NYSCF Robertson investigators. We're delighted to have you. We're looking forward to getting to know you and upwards and onwards. So it's a great pleasure. So thank you very much. And now we are going to hear from several NYSCF Robertson stem cell investigator alums who are working to get us out of this pandemic. And we all hope that they can go as quickly as they can. And they have different and innovative approaches to the research, which is certainly called for. So to moderate this panel, Dr. Kristen Brennand, NYSCF Robertson stem cell investigator alum from the Icahn School of Medicine at Mount Sinai, will moderate today's exciting discussion and set the stage for our conversation. Welcome and thank you.
Thanks, Susan. I'm so excited to be here and to be having this conversation with my fellow NYSCF Robertson Investigator alum. The thing that would make it better if we were all sitting on stage together, able to interact with you and the audience. So these panelists today are a really amazing group at the cutting edge of their fields. And they've all already made significant contributions to their original fields that make them perfectly suited to take on COVID-19 in their labs now. As you know, this is a really challenging disease, unique in so many ways, and it's so important that we work together in ways that we never have before. And so I'm looking forward to this discussion that we're about to have, as the depth of expertise in this group is really incredible, spanning from diagnostics to infection to antibody treatments. And so before we begin the discussion, in order to frame our chat, I'm going to introduce each panelist one at a time and ask them to briefly describe their COVID-related research.
Before you do that, I'm going to sign off, and so you have full screen. Look forward to the panel.
Okay, so first up is Feng Zhang, PhD, who is a core member of the Broad Institute, an investigator at the McGovern Institute for Brain Research, and the James and Patricia Poitas Professor of Neuroscience at MIT, and a Howard Hughes medical investigator. His work provided the foundation for CRISPR-based medicines, and his discoveries continue to fuel the clinical translation of CRISPR technologies. Welcome, Feng. I know you've been involved in lots of efforts around rapid testing, using your diagnostic platform Sherlock, as well as surveillance, including a new app. So maybe you could begin by sharing a little about what exactly you've done and what you're working on now.
Sure. It's really a great pleasure to be back here, especially with other fellow NYSCF alums. It's nice to. It's really a great community and a great family to be a part of. So we've been working on a couple of different projects related to COVID-19 since the onset of the pandemic back in January. So I remember seeing a newspaper article in the New York Times about this COVID-19 SARS-CoV-2 virus that's just emerging, and thinking that maybe we can repurpose one of the diagnostics technologies that we have been working on called Sherlock and use it to provide a low cost and also rapid diagnostic for the virus. And I'm sure you read in the news and probably remember that there was a huge shortage of tests, and also the turnaround for test result is very slow. And so there's even now there's still a huge need and urgency to have increased testing capacity. And so for the past few years, we've been exploring CRISPR protein diversity. And one of the systems that we found, this CRISPR-Cas13 protein, has the ability to recognize a RNA signature. So this could be a virus or a bacterial signature, and then turn that recognition into the activation of a reporter. And so this allows us to be able to have reasonably good sensitivity for detecting the presence of virus or RNA. And so we've been working on turning this technique toward COVID-19. And we initially developed a number of kits for different research laboratories, sent them out, and got feedback that the assay works, but sometimes it can be a little cumbersome to run. And then we turn our attention to making the test more robust and have even higher levels of sensitivity. So then we eventually develop an optimized set of reagents that would provide performance that are comparable to RT-qPCR based assays, and then can give you a result back within about 30 minutes using a fluorescence-based readout. So as we were developing this, turns out that these techniques belong to a class of diagnostics called isothermal reactions. But isothermal reactions still require heating and also some minimal handling of the liquid reagents. And since about April, we've also begun to develop a small, low-cost device that can allow the running of these tests in a turnkey fashion, so that if you can develop a cartridge and load the reagents onto the cartridge, then all you have to do is to add the sample to the cartridge, and then you can insert it into a low-cost machine and then get a readout back. And so we're continuing to work on this. One of the biggest challenges is to be able to scale this, to make many tests to distribute to many use cases. And so that's something that we're continuing to work on. But these are just one of the aspects of the COVID-19 effort that many scientists are working on, and I'm really looking forward to engaging in a fun discussion today with all the other panelists around their COVID-19 efforts.
Thanks, Feng. Now I'll invite Shuibing Chen to the stage as well. Shuibing is the Kiltz Family Associate Professor and Director of the Diabetes Program in the Department of Surgery at Weill Cornell Medicine. She pursued her PhD with Dr. Peter Schultz at the Scripps Research Institute, and then joined Dr. Doug Melton's lab as a postdoc at Harvard University to study the directed differentiation of human embryonic stem cells, which is where we first met. And she's now a leading expert in stem cell-based screens. Shuibing, I know you and your team have been working hard to establish a variety of tissue-specific organoid models to study SARS-CoV-2 infection across cell types, including pancreatic beta cells. And so I was wondering if you could briefly describe the uniqueness of infection and cellular response across the lung, cardiovascular, and nervous systems.
Okay, thank you, Kristen. It's really a great honor to come back and join this panel discussion with Feng, Jay, and Deepta. So we, at the beginning of the COVID, we basically set up a large consortium working, including several labs at Cornell, St. Catherine, and Mount Sinai. We basically derived 10 different types of organoids or cells from human pluripotent stem cells. And then we want to ask 3 questions. The first question will be, which type of cells or organoids are permissive to SARS-CoV-2 infection? And the second question will be, if they are permissive, how they respond to SARS-CoV-2 infection. And of course, the third question will be whether we can adapt it to a screening platform look for the drug candidate that can either protect the cells, like show the antiviral activity, or like rescue the cell survival. So at the beginning, when we developed these 10 different types of cells or organoids, so we see some organoids are very permissive to SARS-CoV-2 infection as expected, such as lung, because lung is a major organ that targets by SARS-CoV-2. We also find like colon organoids are very permissive, like liver organoids are very permissive. So that is because the patient also shows some symptoms like GI tract symptoms or liver symptoms. And another cell type we found are very permissive is cardiomyocyte. And this morning we heard a very nice talk from Dr. Chikmarie about how cardiomyocyte responds to SARS-CoV-2 infection. And it's actually cardiomyocyte are quite special because there's several labs already reported response of cardiomyocyte to SARS-CoV-2 infection. Until now, it's still very controversial whether we can detect SARS-CoV-2 in the cardiomyocyte in human autopsy samples. So there are several autopsy reports say that they fail to detect the SARS-CoV-2 infection. So only positive reports is using an autopsy sample from a patient who passed away 1 day after hospitalization. So suggesting maybe timing is very important to detect the SARS-CoV-2 infection. And two cell types that were very surprisingly to us that are permissive to SARS-CoV-2 infection. One is pancreatic beta cells. And after that, actually recently several studies start to report the new onset diabetes in the COVID patients, suggesting that COVID or SARS-CoV-2 may have some causal effect on the new onset of diabetes. And neuron, we just heard a very nice panel on neurons we can discuss later. But for our effort, we'll work with Lawrence Suder's lab and Tao, and who is also a NYSCF fellow. So we basically test the microglia, cortical neuron, and Doppler neuron, and consistent with what we heard, that we did not see any infection in microglia. We see very low infection on cortical neuron, but we actually see very high infection in Doppler neuron. And currently we are running some screening to basically find a small molecule that either can block viral infection or that can protect different type of cells upon SARS-CoV-2 infection.
Thank you. Now we'll invite Jay Rajagopal to join us as well. So Jay is a professor at Harvard Medical School and a member of the MGH Center for Regenerative Medicine, where his laboratory is interested in modern approaches to study lung regeneration and reframing the cellular basis of lung disease. He performed his residency and chief residency at MGH, followed by pulmonary critical care fellowship, and then a postdoctoral fellowship, again with Doug Melton at Harvard University, which is, again, how Shuibing and Jay and I all first met. So Jay, I know you've been working with the Human Lung Cell Atlas for a number of years, which has quickly pivoted to identify the lung cells that have the COVID-19 receptor and which can be infected. So I was hoping you could please briefly describe your work exploring how age, smoking, and other factors impact lung susceptibility to SARS-CoV-2. We can't hear you, Jay. I think you're muted.
Hi, Kristen. Can you hear me now?
Yeah.
It's great to see you. It's also good to see Shuibing and Feng here. So we got involved with this right at the very outset. I'm a pulmonologist at Mass General, and as I was watching the floors shift into ICUs, the activity on the scientific side was just as remarkable, because all of these folks that were interested in the Human Cell Atlas had formed a sub-consortium for people that were interested in the lung, just as they are in all the other organs. And as soon as SARS-CoV-2 came out, I mean, it was very clear that at least there was some molecular apparatus that was understood about the ACE2 being the spike protein receptor, and then the protease, TMPRSS2. And we just decided very quickly to kind of scan through the abundance of data we had to assess where and in which cell types these two molecules might be co-expressed. And right from the very outset, it was quite interesting, because it seemed like in the respiratory tree, goblet cells and ciliated cells in the nose were the highest expressors, which kind of made some sense in terms of potential transmissibility of this virus. And then as the consortium started looking deeper, it became clear that the type 2 cells, the one that produced the soapy substance in the alveoli of the lung, co-expressed this receptor, and probably the site for infection that produces pneumonia. But as we continued to go on, the cell types just started to make sense. Enterocytes of the GI tract were double positive, which suggested this might be an explanation for the diarrhea-like symptoms. Corneal epithelium was positive. The special sustanticular cells of the nose, which is associated with this inability of smelling in COVID-19, also came up. And then what was most amazing, actually, was at the end, 107 different studies came together from groups around the world, many of which we had not even heard about before. And one of the nice things for this group is 16 of the 22 lung data sets that we produced were completely unpublished. So people kind of figured out how to get their data to a central source and protect that data, and started mining it. And then with that kind of huge power, where we had, you know, over 1 million cells, we were able to actually correlate expression to kind of clinical findings. So the things that came up were epidemiologically age, gender, and smoking status all seem to affect the course of COVID-2. And all of those things started making sense with respect to the receptor and ACE2 expression in the lung. So, well, for example, you know, men have more ACE2 and TMPRSS2 expression in the airway. Males have more. And interestingly enough, age was really a profound variable. And although the embryo has a lot of these molecules, soon after you're born, between the age of like 3 months and 3 years, your expression goes way down. So we were able to pretty rapidly generate hypotheses about some of the epidemiologic characteristics of COVID-19, which, you know, hopefully we can verify over the course of time.
Thanks, Jay. And now I'll invite our final panelist to the screen. Deepta Bhattacharya is an associate professor at the University of Arizona. He completed his PhD at UC Berkeley and his postdoc fellowship at Stanford. His lab studies both stem cells and antibody responses to infections and vaccines, and so he's probably the most obvious expert here on the panel. Deepta, could you share with us some of the work your team has been doing towards antibody responses, durability of protection from reinfection, seroprevalence studies, and the potential of vaccines?
Yeah, and as you say, it wasn't actually very much of a pivot for what we were doing anyway. I mean, the lab has been long studying acute RNA virus infections, and so you know a lot of the framework was essentially already there to do some of this work. So, you know, what we were really interested in is there something, you know, what are the unusual aspects of the immune response to this virus? And what are the things that are actually pretty normal and conform to all the things that we've learned over the past however many decades of viral immunology? And so what's clear is that a lot of things can go wrong very early in terms of the innate immune response to the virus. Lots of things happen. So really uncoordinated response that can happen in the subset of people that get severely ill. What we're really interested in is, first of all, you know, understanding how the subsequent adaptive immune phase functions. Is there lasting immunity? And, you know, what does that actually project out in terms of optimism or lack thereof of vaccines? And then also using some of the same tools to get a better emphasis or a better estimate on what is actually going on in our local community. So when we first started this, you know, I think we started getting ready for this in probably late February, early March, when it became pretty clear that the pandemic was not going to be just confined to a few geographic regions. It was going to get everywhere. And so at that point, we started to get ready for it. And there's a lot of speculation, you know, how much pre-existing immunity is there from other coronavirus infections? What fraction of the population has already seen it, might already be immune? And so I think the goals we had was, first of all, to develop a very accurate serological test. It's gotten a lot of heat in the media, for rightfully so, because a lot of them are wrong. And so I think one of the real mandates we had is like, okay, we can't get that wrong. We can't tell someone that they're antibody positive and likely immune when they're not. And so it was a matter of developing, you know, orthogonal tests to make sure that the things that we actually said were right. And then taking those subjects that we found from statewide surveys and understanding what the course of the immune response does. And so thankfully, what we've really been finding is that even though things can go badly wrong at the beginning in a subset of people, you know, the durability of the antibody response looks pretty good. And so I think that there is some initial concern that everything crashes down to zero. And I think maybe this is something we can get into a little bit later, is that I think, you know, lots of people wanted to jump into the effort and contribute in what ways they could, sometimes without the background knowledge of what a normal immune response does, which is it rises, it falls, and then it sort of stabilizes. And the question is, you know, was it going to stabilize? And basically, that's exactly what we found. So there seems to be nothing particularly unusual about immunological memory and our ability to form it. So that bodes well for vaccines, in that even though there are some cases where we've been able to generate effective vaccines when the immune response to the natural infection isn't very good. Say human papillomavirus is one great example of that. Most of our successes have come when the normal response to a natural infection leaves you immune. And I think that again, I think it bodes pretty well with the prospect of getting an effective vaccine.
Well, that's great, everyone. I think it was a really good start. And so to get the panel discussion going, I'll start with some really easy questions. This first one is maybe more towards Shuibing and Feng. When and why did you each decide to pivot your labs into virology and COVID research?
Maybe I can start. We decided to work on COVID back to around January, and it's actually a very personal reason. As we all know that COVID forced the break in Wuhan in China, and it was around Chinese New Year time, and my mom was sick and was hospitalized. And I really want to visit her, but the whole country was shut down. So I was really sad, and I think maybe we should do something to this virus since we worked on Zika before. So that's the reason we did start working on that. And then, as we all know, that New York City unfortunately became the center of the pandemic in March and April, and then we continue working on that. Yeah.
Mm-hmm. Yeah. When for us, it was very much the same. When COVID first broke, there were a lot of people who got in touch with me, asking me whether or not there is a easy way to apply Sherlock for detecting COVID-19. And then also as the severity of the pandemic become more and more apparent, and also as the need for testing becomes more and more urgent, that just motivated us to work even harder to try to get more testing solution out there into the community.
Are there any specific ways for any of you guys that you think that the work that were doing, and I guess this is again really obvious for a few of you, but how were you well positioned to pivot?
Is that to me, Kristen?
Sure.
Yeah, I mean, like I said, it was not much of a stretch because an awful lot of what we've done has been working on acute RNA viruses. And we've done a lot of work with flaviviruses, Zika, dengue, West Nile virus, Japanese encephalitis virus. We've also done some work with flu. And so a lot of the technical know-how was already, built into the system for what we do. I think, where we did pivot is obviously, the specific virus we were working on. And I guess what we found is that there was just so much interest from everyone in getting this going, is that in unprecedented ways, I mean, the university leadership and all sorts of people around our community, pulled us all together, and, we were able to do things at a scale that certainly we were never able to do before. So, setting up testing sites all over the state, and all the investment that went into it. So I don't know that it was topically that much of a pivot, but certainly just the scale and the magnitude in which we were doing some stuff was very much different than anything we'd done before.
I think that's a great segue to my next point, which was going to be, how has working on COVID-19 changed your science?
I'll start. I think COVID-19 changed our science quite a bit. It went from in-person and a lot of close interaction to many of the interactions taking place over Zoom. And I think as we kind of adapted to this new mode of working, we then we figured out ways to make it work as well as it can. So adapting a number of different online tools, Slack, different kinds of messaging or data sharing platforms to really make the communication as seamless as possible. In terms of the actual science, I think the pace at which the overall scientific community have coalesced and collaborated to make rapid progress around COVID-19 is really inspiring. And we try to help other people as much as we can for what they're trying to do. But then at the same time, we try to see if there are any ways that we can collaborate with other people. And I think it's really been one of the most collaborative periods of science that I've participated in. Almost anyone that you reach out to have a conversation or to have to initiate a collaboration is always received with warmth and enthusiasm. And that I hope it will continue and not just end with as the pandemic finishes.
Yeah, I agree with what Feng said. It's really change the way we do science. We definitely do. Previously, a lot of lab, we have our personal interests. We focus on the area. We have collaborator, but it's more like a certain topic. And COVID really push us to basically collaborate together to use our own expertise and move the field forward. I can share some personal experience. For example, when we do the screening, we need to get pseudovirus from David Ho's lab in Columbia. And the OBSL3 work need to be done in Bentandover's lab at Mount Sinai. So the way we run it, we grow organoids, we send it to like Mount Sinai to run infection, and we get RNA-seq data, and then we analyze the data. So previously, it will be a little bit challenging. This kind of collaboration may take like several weeks or even several months to set it up. But when the COVID comes, people immediately start working together to move it forward, which is really impressive.
I totally agree. And we did not move in, I think, as fully as your labs did, but it's maybe more fearless in my science that I had put myself in this psychiatric genetics pigeonhole, and realizing that we have tools that can spin has really challenged me to think about what the next questions will be too.
Yeah, I can echo a lot of the things that people said. I mean, we had started building this consortium, but now, we're well represented in Asia, in Europe, in North America, in Australia, and it was surprising how easy these connections were to make. And I think I mentioned earlier that a lot of the data that we assembled was unpublished data, and people weren't the slightest bit hesitant about it. Their institutions made it facile to accumulate this data into a central repository in Europe. And I've just never seen that kind of incredibly rapid sharing before. So it was really exciting. And I got to say, I mean, a lot of the postdocs, the hospitals, fortunately, and I think a lot of the labs were wonderful in that they permitted this one form of work to go on, even though it was difficult to allow people to work. And I know that's true for all of your institutions. So the COVID work was just able to move on. And I've got to say, the young people, the graduate students and the postdocs and the computational biologists that I was watching, they were working round the clock. I mean, my email was just full of data at every possible hour of the day, given the kind of global nature of the response. And I think all the young people that were working on it were really inspired about the way science was happening also.
I think while all of us scientists have been coming together, the country has actually found itself more and more divided over whether this pandemic is over and how we should be responding to it. And I think it's forced us to reconsider our role, not just as scientists, but as community members. How has your COVID work changed the way that you feel about scientific communication with the media and with the public? And what are the consequences of not doing it well?
Yeah, I can maybe, I'll take the lead on this because I can, from my own experience, and I've done, some degree of scientific communication with the public before this, but it was almost always like a primed audience, people who are already engaged, very interested, not really lay public. And I think that the way that you can communicate with an audience like that is very different than what it is right now, where everyone is just, doom scrolling all the time, looking for what exactly is happening and what the scientists are saying. And from my perspective, particularly as primarily a viral immunologist, I got called into a lot of media interviews and things like that. And, how I initially first approached it is exactly the same way that I used to do it in the primed audience, or even when I'm really just communicating to other scientists. And I guess what I learned very quickly is that that's not working quite right. And I think in some ways, maybe it exposed some things that maybe we need to reconsider in how we communicate even to each other. So, I guess the simple formula is for me, tell people what you think and why you think it, and I think that there's been a lot of efforts that, just as part of how we communicate with each other as scientists, we almost maybe play up the uncertainty a little bit too much. You take something that might be wrong and you say, well, I don't really know. and you emphasize the I don't really know part without actually giving the context of what you do know. And as scientists, that's okay. You know, we understand what that means. But when you do it with the public, what we found is that that really doesn't fly quite right. So let me give you an example of something near and dear to my heart, right? So at the very beginning, there was a lot of question as to how long immunity lasts, right? And so the standard answer is like, well, we don't know how long it lasts. And so then what came is like a lot of maybe poor quality information that then filled the void and saying, well, oh my God, immunity lasts a few weeks or even just a couple of months. So instead of saying, we don't know how long immunity lasts, what I've come around to saying is like, well, we don't know how long immunity lasts, but if it's anything like the first SARS coronavirus, it will last for at least a few years. And so I think, giving people that context of what we have learned and setting the range of uncertainties around that, like in a sort of a Bayesian way, is I think really the right way to do it. And if they get the wrong impression first, if they get bad information first, that becomes imprinted and very hard to undo. We've seen that with face masks. We've now seen it with immunity, which I think will have effects on vaccine uptake. And so again, I think the real key thing is just to treat people like adults, tell them exactly what you think and why you think it, don't overhype it, and don't over uncertainty it. I think that's sort of the lesson. Those are the lessons that I've learned.
Yeah, what I learned is that because we run.
Oh,
go ahead,
Shuibing.
Yeah. Okay. So yeah, what I learned is because we run a lot of drug screening, and I was always asked by lay people, I say, when do you have a drug? Okay, so you guys screen so many drugs, which one work? Can you recommend someone we can use? So I think that's very important to get, because people are so eager to get this problem solved. And if we have vaccine, if we have antibody treatment, if we have chemical drug that can work to treat the even severe case of patients, our life will be much better. So my feeling is it's very important to give people a full picture and to try to educate the people to understand the drug, at least for us, the drug developmental process. And every time when we try to explain that, we will say, although we read a lot of exciting news, say this drug may kill SARS-CoV-2, but many of them are either from a cell culture, it's not human experiments, are either from animal experiment. But before we move to the real clinical drug, we claim it's have activity, it need to go through phase 2, phase 3 clinical trials. So in that scenario, after several times, and the audience start to realize it's a long-term way, it's a long way to go to find the solution. It's not really one shot, but one sentence can answer the question they are looking for. Yeah.
Jay, did you want to?
Another interesting thing that came up, I think, both on the clinical side and the research side, was this use of very early preprint publications of what were initially almost anecdotal evidence. You could call them trials, but they were so small that they couldn't possibly have statistical relevance. But they kept getting dumped out onto preprint servers, and then the media immediately caught that bug from any one of these studies. And it was remarkable to see even academicians around the world kind of suggest hypotheses that needed to be pursued because there was nothing else to do. So even though there was kind of very limited data, things were being done simply because there was a suggestion that something might be true. And a lot of those therapies, interestingly, are known to be toxic in one setting or another. You can do harm with those things. But there was that desire to do something. And there was this interesting kind of discussion, I think, in the entire community. And I think most people felt if we could move along science a lot faster than we normally do, we'd be doing that. The thing where I thought science really moved along a lot faster here was the sharing amongst scientists. That moved faster than possible. But communicating with the general population that certain things take time is a difficult thing to do when everyone feels so pressured. But I agree with Shuibing, you have to repeatedly get across the idea.
So we've got about 12 minutes left in the panel, and I want to remind the audience that they can type in questions in the chat bar. But while we're waiting for them to do so, let's move into some harder topics, perhaps the only ones really matter, right? How long till things get back to normal? What's it going to take for us to get there, and what will it look like?
Deepta?
Yeah.
I was going to punt to Feng first. Yeah, let's let Feng. I'll let Feng go. Let me tell me my thoughts.
Well, I think what is clear is that people are working very hard to try to develop testing solutions, develop vaccine, develop antibody therapeutics, and everyone is working as quickly as possible to deliver these possible solutions to address the pandemic, the health tolls, and also try to be able to allow us to open society sooner. Exactly how long this will last, I don't know. I think my guess is as good as your guess. Sounds like some vaccines will be ready early next year, but larger volume production of vaccine won't be ready until late next year. And hopefully the vaccines will be effective and the immunity will last for a long period of time, more than just 3 months, as reported in some cases. I think all those things will play into this sort of shifting time window for how long the pandemic will last. But I think what is good is that I think people are becoming more cautious. More people are wearing masks to follow the public health advisory to limit transmission of viruses. I think as the winter comes and as we see case numbers rise, it's going to be challenging. But I think many people are recognizing that and then trying to follow the best practice possible to limit the risk that they face and also the risk that they may pose to other people. So I think by sticking together and following the latest science and trying to make sure that we do the best we can, we'll get out of this sooner than if we don't. And hopefully, I'm sick of doing Zoom meetings and Zoom panels. It'd be great to not have to do Zoom anymore. So I'm definitely very eager to get out of the pandemic.
Yeah, I mean, I think that one of the things that makes it just so hard to predict on how long it's going to go on for, and how whether it'll come down fast or whether it'll come down with a slow decline. I mean, I think that for me, as a basic scientist, one of the things that has been really eye-opening is just how much human behavior affects the things that honestly, the countermeasures that we could have taken that would effectively make some of these interventions we have essentially like ancillary. We look at countries like New Zealand, South Korea, Japan, Taiwan, Germany, and what you say. I mean, I just am incredibly jealous, to be honest. If we were in New Zealand, we wouldn't need a vaccine. It wouldn't be a relevant question as to how long antibodies last. And I think that what this is really exposed is some of the societal problems we have in terms of inequities, in terms of healthcare, in terms of people not really feeling like they can take a day off even if they don't feel well. These are really long-lasting things that we have to address fundamentally. Because we're in the midst of it, I think the pandemic's going to take a little longer than really it should. On the optimistic side, again, getting back to the immunology, I do think that, from my perspective as an immunologist and virologist, that this is not an especially complicated virus to vaccinate against. I am pretty confident that more than one of these vaccine platforms, including Moderna, one of our alums who started that, are going to be, likely to be at least partially effective in mitigating disease. And then some of the later ones will probably actually give a substantial reduction in transmission as well. And so in that regard, I do feel like there are options coming out there. But again, we have to build trust with the public and convince them that this is the right thing to do. And there are just so many challenges that go beyond just the science that I think that are really critical things to address to bring down this pandemic.
Jay and Shuibing, when we were in Doug's lab together, I remember there were times where people would talk about diabetes being an engineering problem rather than a biological problem, that it would be solved by pumps and testing first. And I wonder, listening here, if again we have an engineering problem rather than a biological one. There will be a vaccine. We don't know how good, and we don't know how many people will take it. We don't know how often they'll take it. But testing, that could be our way out. You can have a wedding when you can rapid test everybody at the door. You can have a school when you can rapid test every student each day. What do you think, especially you, Feng? When are we going to have affordable home testing that allows, if not daily, at least event-based testing? Is that something that you think is going to be around the corner?
I think it will have to be the combination of many different approaches. So there's going to be centralized laboratory testing, and there's going to be the rapid antigen-based tests, and then there's going to be more sort of home-based tests that use molecular detection for RNA and DNA. Any single one of these will probably not be enough, but I think there are a lot of groups working on bringing increased capacity of different types of tests, and I think together that's what's going to make it possible. When it comes to the cost issue, I think one of the things that will probably have to happen is pool testing, because testing a single sample will be at least $10 or maybe $15, and the only way to bring it down to $1 per person per test is to pool. And as long as the prevalence of disease remains low, say 1 in 100 person or less, then you can pool economically, because most of the pools you want most of the pools to be negative, because if every pool is positive, then you have everybody anyway. So I think we're making a lot of progress, and there are good pooling options that are just starting to emerge, that are going to be using school systems and in offices. So I think hopefully in the next couple of months we'll see a lot more capacity.
For the MDs on the panel, what therapies do we have now that weren't available or used in March? What should we be on the lookout for? What's coming to the pipeline?
Yeah, so I think clinically, I think when I talk to my clinical colleagues in the hospital, actually, they think that the pre-ICU care has actually substantially improved, and that they're able to keep people out of the ICU from getting sick more quickly. And I think remdesivir has made a difference. And the other thing that was a big surprise was steroids, because by and large, what COVID-19 seemed to do was produce this constellation syndrome called ARDS. And it is a good lesson. I mean, the teaching in ARDS is no matter what caused it over time, steroids were unhelpful. But for whatever reason, and we still don't really understand it, steroids do seem to be helpful in COVID-19. And I think for a lot of people, that was a big, big surprise. I mean, almost all pulmonologists and critical care people I knew would have said that trial was barely worth doing because it's always failed. So I think that's actually improved. And so I think that there's going to be, even with a surge, there'll be patients that are certainly admitted to the hospital, but I think we'll be able to keep more of them out of the ICU. And that should help in terms of the mortality figures. And it should also help in principle with just hospitals getting overwhelmed. Because I saw that kind of firsthand. I was on those Zoom meetings also. And literally, like, every room at Mass General was converted into an ICU. It was really amazing. There were pediatricians taking care of adult patients with a ventilator. And they'd never been trained to do those kinds of things. So I think, although that's not a cure, just the ability to manage those patients better medically is going to help the hospitals just survive kind of periods that are worse than we want them to be. And the other thing I would say, Kristen, about that's interesting, and I think this speaks to what Deepta was saying about it being a behavioral disease. I've also been impressed by how little nosocomial in-hospital transmission of this infection there has been. I mean, I know very few nurses or doctors who have gotten this disease despite the exposure. So masks and the other preventative measures work. And I think, so education on that, I think is also, I don't think a vaccine alone is going to do this. First of all, people have to take the vaccine. So I really think, unrelenting education is going to have to be part of this. And as Feng said, I mean, people are getting it. As things continue to happen and the media keeps displaying things, I think people are slowly getting it. But I think our community can't relent on the fact that, masks are going to be as important as a vaccine, I think.
No, I agree. The messaging from our hospital has been that masks are 99% effective, and a vaccine could very well be 50% effective. And so we should all choose masks right now. I'm seeing one more question here. How do we recommend talking to people who have a profound mistrust of science then, to the point of these elaborate conspiracy theories, and who won't even trust good quality and reliable data? What do we do to help here?
I mean, I think, one very key thing, and I've learned this from the trenches of talking to people who, are deeply skeptical of vaccines, is that shaming does not work at all. And so I think it's very much a bad idea to get into shouting matches and tell them how dumb they are. You don't want to go there, right? I think the first thing is to sort of acknowledge where the source of their mistrust and concerns come from. And, to be honest right now, I mean, there's a lot of surveys out there that say that people, if a vaccine comes out, then a third of the people wouldn't take it. And I think, for me, when I hear that, I think one of the first things you want to do is acknowledge, well, I wouldn't take it either until I've seen the data. You know what I mean? And so you say, like, yeah, I mean, you're basically giving a small bit of ground that they may have a slight bit of valid concern about something, and then you use that to sort of nudge their way forward and saying, when the data do come out, pay attention to the people who have got no skin in the game, and basically just keep sort of like acknowledging that there's at least some basis to something that they may be worried about. And then also, I think really making ourselves human in this too. And we've all been affected by this in some way or another, right? And trying to form those individual connections, I think, is really important. That's the way I think that you start to get them to at least, if not fully convince them, to maybe not shut the door quite so hard.
Well, this has been a really fun panel, but I see that we're already out of time. Is there anything that you guys want to add before we close and invite Susan back?
Just want to say, hope everybody stays healthy and stay well. And if we hang together, we'll get through it.
Yeah.
Yeah.
Thank you, guys. I mean, this has really been incredible. I'm thrilled that you are all working on this. I just can't wait to stop all the Zoom meetings and see everyone together. It would really be wonderful. So thank you for everything. Thank you, Kristen, for moderating.